Let's discuss the dairy cow feeding physiology part 3 with the glorious name of Volatile. In part 1 and part 2 we discuss the digestive tract anatomy of a cow, human dynamics of a cow, human fermentation or metabolism of carbohydrates, fat and protein and nitrogen. As well as we discuss the milk urea nitrogen normal levels and their indication. Today in this video we will discuss some mineral and vitamins role, volatile fatty acid production and factor impact on Rumen pH. Part 1 and part 2 link present in the description. You can click on that links and go to the part 1 and part 2. Minerals and vitamins. Minerals and vitamins have an important role in human microbe growth as well as in the cow growth, reproduction and production. As we know, vitamin B family, especially the vitamin B12, synthesized in the rumen of a cow. And cobalt is a necessary mineral that needed for the synthesis of the B12. If we fed the cow cobalt not according to their need, then vitamin B12 not synthesized in the body as per need. If we fed cobalt at a proper level to the cow, then there is a proper synthesis of vitamin B12 inside the rumen of a cow via rumen microbes. Likewise, cobalt, sulfur has also important role because rumen microbes made a microbial protein, and the protein are made of the peptides and amino acids. And some amino acids are sulfur containing. That's why sulfur have an important role in the feed. If the feed is a sulfur deficient, rumen microbes are unable to make the sulfur containing amino acid. Ultimately, there is a less production of the microbial protein. And in the feed, nitrogen to sulfur ratio is 10 to 1 or 12 to 1. Optimizing human digestion, the volatile fatty acid production. Volatile fatty acid production take place in the rumen, and there are mainly three types of volatile fatty acids that produce inside the rumen: acetate, propionate and butyrate. While valerate and isobutyrate are also produced inside the rumen, but their proportion of the production or their ratio is almost near to zero. That's why they are neglectable. Volatile fatty acids are a main source of energy inside the body of a cow. Acetate are the 2-carbon volatile fatty acid, and their proportion is, or their percentage is 55 to 70% of the total volatile fatty acids. Acetate are made up of the digestion or fermentation of the structured carbohydrates. Acetate volatile fatty acids goes to the milk glands and form fatty acid there. As well as acetate volatile fatty acid used in the tissue as a source of energy. Second one is a propionate. Propionate is a 3-carbon chain. Propionate is made up via the fermentation of the starch, sugar and via the pectin digesting bacteria. Propionate consists of 15 to 13% portion of a total volatile fatty acids. Propionate used in the liver as a source of the glucose. That's why propionate called the glucose precursor as well. While in the milk, propionate made the milk lactose. The third one is a butyrate. Butyrate is a 4-carbon chain, and it consists of the 5 to 15% portion of a total volatile fatty acids. Propionate used as an energy source in the tissues, as well as propionate made the milk fat. And acetate to propionate ratio in the rumen is 2.2 to 1 or 3 to 1. First of all, what's the normal pH of the rumen? That we discussed already in the previous 2 videos, that the normal pH of the rumen varies from 5.5 to 6.8. While in upcoming slides we will discuss that the rumen pH will be ideal if it will be around 6, because the rumen pH will be 5.5 to 6, then it's good for the starch digesting bacteria. Or if the rumen pH will be 6 to 6.8, then it's good for the fiber digesting bacteria. That's why rumen pH will be 6 for will be 6 considered ideal, because it's a good for the both type of the bacteria. And keep in mind, if the rumen pH will goes towards 7 to 0, then it will be acidic. But if Rumen pH will move from 7 to 14, then it will be basic. 7 is a neutral point, or we can consider it a base point. If pH moves towards 5 even, then it will be considered acidic. But if pH moves towards the 8, then it will be considered basic. Rumen pH range is 5.5 to 6.8 as discussed earlier. 5.5 to 6 is good for the starch digesting bacteria, while 6.6 to 6.8 is good for the fiber digesting bacteria. And Rumen pH will be ideal if it's on 6, because it's good for both starch digesting bacteria as well as fiber digesting bacteria. And factors impact on the Rumen pH. First one is forage to concentrate ratio. If there will be higher forage inside the ration, there will be higher pH, because the 6 to 6.8 pH is favorable for fiber, or is favorable for fiber digesting bacteria. There will be the more production of the saliva, and if saliva will produce more, then there is more bicarbonate ions will come via saliva in the rumen, and the rumen pH will move towards the basic, that is ultimately 6 to 6.8. And if there is more fiber in the diet, there will be more acetate production, and if there is more acetate production, ultimately there is a more milk fat in the produced via cow. We already discussed the thumb rule. If we fed more forage to the cow, there will be more acetate production, there will be more fat. But if we will fed more concentrate to the cow, there will be more propionate production, then there will more be milk production. Likewise, if we fed the concentrate beyond the higher limit to the cow, there will be a higher propionate production. But keep in the mind, there will be a decrease of the pH, because the concentrate is some kind of the form of a starch, and the starch digesting bacteria, their ideal pH will be 5.5 to 6. That's why there will be decrease in a pH. If there will be a decrease in a pH, animal moves towards the acidosis. In case of the higher concentrate beyond the limit, there will be reduced intake. Ultimately, there will be reduced microbial nutrient production, and there is a milk fat depression while milk protein increase in this case. Life is the name of a balance. So in a rumen, there will be a balance in a forage or the concentrate. Ultimately, there will be a balance in the cows or in the ruminant diet in forage to concentrate ratio. Second one is the physical form of the feed. Grinding, pelleting, chopping or mixing, they have a crucial role in the rumen pH. If the particle size is too short, first of all discuss the forage mat. If the particle size is up to the mark or up to the standard, then the large particles float on the rumen or on the ruminal fluid. These particles stimulate the rumen and the esophageal groove for the regurgitation process. But if that is the forage mat. But if the particle size is too short, cow will consume less than 2.5 kg of the particle over 1 inch. Then forage mat will not made inside the rumen, because there is a small particles, and the small particles float on the lower portion of the rumen. Forage mat will not form inside the rumen, then the regurgitation process will not occur. There will be not proper fiber digestion, and ultimately there will be lower pH. Second one, if we offer the cow to the concentrate finely grinded particles, there will be more starch exposure, more starch bacteria acting on it, ultimately less pH below the 6. Just like this, stream flaking or pelleting or grinding cereal grains increase the rumen starch availability. That will lead to the acidosis as well. Third one is the feed intake. If animal will intake the feed beyond the higher limits, then there will be more carbohydrates inside the feed, more concentrate inside the feed, more substrates available for the starch digesting bacteria. There will be the lesser in the pH, there will be a decrease in the pH below the 6, ultimately leads to the acidosis. Fourth one is the moisture content. Keep in the mind, always saliva have a role. No doubt saliva have a bicarbonate ions, they maintain the Rumen pH, but saliva act as a lubricating agent as well. They swallow the feed from mouth to esophagus to rumen. Saliva have role inside it, but if feed is already wet, saliva will not produce according to the normal level. It's the process of the homeostasis. Wet feed can reduce the rumen can reduce the saliva production. If there is the less saliva, there is less carbonate ions production, and there is less carbonate ions production, then ultimately there is a decrease in the Rumen pH, because we know inside the saliva there is the bicarbonate ions that maintain the Rumen pH, because bicarbonate ion have a neutral effect on a Rumen pH. Bicarbonates ion can increase the Rumen pH 7 to onwards towards the basic. If there is more than 55% moisture inside the feed, then there is a less dry matter intake due to the less production of the saliva. The fifth one is unsaturated fats, already discussed in the part 2. Because unsaturated fats present in the vegetable oil and the seed oil, they have the negative impact on the fiber digesting bacteria, as well as the unsaturated fats cover or made a layer around the fiber. Due to these, fiber digesting bacteria unable to act on the fiber. Ultimately, there is a decrease in the pH. Just like that, the grinded when grinded seeds rupture, their cell wall exposed, more carbohydrates are exposed, more starch digesting bacteria act on it, ultimately leads to also towards the acidosis. The sixth one is the method of the feeding. It's also depend upon we have fed to the cow, the TMR or just the concentrate, and these are all the factors that decrease the Rumen pH, that leads the rumen towards the acidosis. If you feed more corn silage to the cow, lead to the acidosis. Just like that, if there is a decrease the forage particle size, due to this, they will not make the forage matter inside the rumen. There is a less digestion of the fiber, ultimately lead towards the acidosis. Just like that, if there are more carbohydrates inside the feed, more starch digesting bacteria act on it, also leads towards the acidosis or a lower pH. And if there is an increased Russian moisture, ultimately less dry matter intake, less production of the bicarbonate ions, ultimately that also leads toward the acidosis. That's all. Thank you so much.